Good morning. Thank you very much for the invitation to come and speak here. Fantastic location, and indeed a hotbed of technological development. I'm really excited to be here so I can share some of my experience with you about mastering immunity. And as far as the unwanted immune responses to biotherapeutics are concerned, this is definitely the kind of mastery that can take many years, but is also best done with guides who are experienced and experienced in failing in places as well. So during this symposium, we'll come across some of the failures we've had, but that we can really learn those lessons. So what I've talked to you today is about the risk assessments that we do for immunogenicity. And to put into context how we might go about integrating the data, running some simulations, and then really working towards what does a successful product launch look like. So we would all love to have no immunogenicity, and I don't think there are any products that have absolutely no immunogenicity. So we have something we live with, we just try and manage the risk and mitigate that. Oops, press both buttons at the same time. Oh, there we go. So I want to take you through some of the functions of the immune system and the risk factors, and then to look at how we use those two components to understand how to make a risk assessment. And of course, we use lots of assays. So Promian very experienced in working with these assays. I'll briefly touch on these and how we can go about using those. And then go into some of these early IRA, immunogenicity risk assessments, and look at some of the case studies where we can actually use the assays to make some very clear decisions on projects in the early space. So the whole idea here is we don't wait until we're heading to the clinic to think about what's our risk going to look like, but we're proactive in reducing the risk before it gets realized in the clinic. I'll go on to look at some of the systems we've looked at to try and integrate this in silico and in vitro data so that you can get a better idea of what will happen in the clinic. And then I have a couple of slides at the end on should immunogenicity occur, and it's something that is too much for the product class, what you can do in the clinical space. This really is last resort though, so always best to proactively deal with the immunogenicity as you're making your product in the first place. So if I look into firstly the immune system function, so we do try and split the immune system into the three main cell types, so the dendritic cells, the T cells and the B cells. So the dendritic cells, also the antigen presenting cells there, will be taking up our therapeutic. There are ways you can minimize some of that, and understanding things like glycosylation patterns can help reduce that, but there will always be some uptake. So these cells are then processing the protein into the peptide, so I've got those as the red and green circles, and where they're presented to the T cell on the MHC molecules, this is a really key piece of how immunogenicity occurs and becomes a strong response that we're not looking for. And what's also happening at the same time the antigen presenting cells are presenting to the T cells, the B cells are also taking up the drug recognized through the B cell receptor, which will go on to become the anti-drug antibody. And where the same peptides are presented to the T cells, this is what generates the active cycle of maturation. So you go from an initial, more natural antibody through to a specific anti-drug antibody. So this whole process, you appreciate this is a simplification of what goes on. It obviously occurs in the body, in the lymph nodes, so the germinal centers is where we get the maturation of the ADA. And really our whole job is to try and understand how can we prevent the right-hand side happening because we understand the components in the middle here. So the other piece of information that's really helpful, and I've taken this from the FDA's guidance on immunogenicity assessment, is all the different risk factors that are involved. So this has come already from many years of experience and holds the test of time, that we can look at product-specific factors and then the patient and disease-related factors. Now, usually for the projects you're working on, you already know who the patients and what the disease is, and they won't change. So the one opportunity you have to really change things is to look at your product. So what can you understand about things like the primary amino acid sequence? Quite a few pieces in there about the quality of the product. So what are those critical quality attributes? So we would all love to have perfectly well-behaved monomeric drugs with very favorable lack of aggregation, nice glycosylation. And again, it's something that is probably aspirational, and you have to assess the slightly imperfect product that you have as you go along. One big thing we can do, though, is the sequence, and work on the sequences of our drugs in order to minimize the risk that we're facing. So when we look at the immunogenicity risk assessment, we have quite a few tools to look at aspects of this immune response. I think, as I mentioned, the ideal position is that you start early with the risk assessment. So there's all of the different aspects of development where you can apply the risk assessment. I want to give you the message that the project start is really key. This can allow you to assess what do my risks look like and how hard do I need to work to reduce those. And then the other piece I've highlighted is the candidate selection here, where when you're looking at your options from your lead molecules, which one do I want to take forward, how can I differentiate those so I know I'm picking the one with the best immunogenicity profile? So the other thing that becomes quite clear is that although there are regulatory needs to make sure we have safe products, a lot of products are failing due to the ADA clearing the drug and essentially removing the efficacy. And whilst this is a concern for regulators, it's not necessarily the highest of the priorities as opposed to the safety need. But from a business sense, this is where the most impact comes in and why it's well worth investing in these assays that we'll be talking about in these next 2 days to reduce that risk and understand the profile of your product. So I'll go quite briefly through the different tools available. You will hear a lot more about these over the next 2 days. These are kind of roughly in the order in which we would look to apply them. So when you're looking very early on, you may have in an antibody project many hits, maybe a phage display library you're looking at. And you can use the in silico T-cell epitope prediction software to identify kind of differences in the different hits you've got and look at whether you can identify already some that carry lots of risk. For example, I've worked on a project where we had all of the 6 of the CDRs of our antibody were very nice, strong epitopes. Wonderful if you want to make a vaccine, terrible if you want to make a biotherapeutic. So we go further in this and you can kind of confirm, so we come to the bottom piece here on the MAPS assay. So ProImmune have this as a ProPresent assay, where you can put your protein into those antigen presenting cells and see which portions are presented. So giving you confidence about which of the potential epitopes will be shown to the immune system. As we go into the middle here, we can look at the relative uptake of the drug. So this may be a little bit quicker to do than the ProPresent and not need so much technical input. And you just get a feel for if you have drugs that get taken up more easily. So this can occur if you've got the target is present on an antigen-presenting cell, that depending on where you bind, you get differential uptake. And we go on and look at these in different sorts of T-cell assay as well. So the top right panel on the DC-CD4 proliferation, which I think is ProCern for proimmune, and PBMC assay at the bottom in the middle, which is a ProMap proimmune assay, where you're trying to get an understanding of how much is the drug going to cause proliferation of T cells. So the one at the top right is useful for your final set of leads to understand which candidate may be best to take forward. The one at the bottom middle is really helpful if you've got a range of different potential liabilities and you want to check sequences for sure if they're going to cause stimulation of T cells. Now, an area that we're a little bit slower on in development is the B cell assay, so looking at stimulation of B cells. So there are assays available where you can look at, again, if the B cells are being stimulated with your different proteins, so you can compare those and pick the ones of lower risk. What we're still lacking is a whole immune system, in vitro immune system. There are some systems that have been reported, but these aren't widely available, and it's still unclear about how translatable they would be. So this really would be the key tool set that you can use in order to understand and mitigate risk in your programs. So thinking about how that might be put together as a cascade, some organizations would like to follow this, production line. Which assay do I do when and what order? This is an example, and I want to stress that in a strategic approach, there may be programs that fit very nicely into you do all of this work and you end up going from a higher risk to a lower risk, and therefore it's going to be a lot easier to go through your clinical program. There are also projects you'll work on where you don't need to do all of this. If you already have lower risk and you're able to show by your in silico that there's very little going on, there's no need to go ahead and do all of these other assays. But there may be cases where you can pick examples of an assay to run that's going to help you with a specific decision. Just a slide here to highlight all of the risk factors we talked about. This slide tries to put them into this product, patient and treatment areas. And then I've highlighted what we're looking at directly with the assays. So with the in vitro assays, we're looking only really directly at the product and the specific molecule and what's in that molecule at the time. So again, we tend to do these quite early, so it won't necessarily be clinical material. So we won't know for sure all aspects of the CMC. We also look at a subset of patients or of donors in the assay. So we don't know for sure these are going to be the same as your clinical trial subjects. So quite a lot of the areas grayed out. And I use this slide to try and help us understand that the in vitro assays are not giving us a precise prediction of what happens in the clinic because we're missing quite a lot of the factors. But it does help us very much in understanding the product, and that's really the key. We're reducing our product immunogenicity risk, and we can understand that in the context of these assays. Okay, so some case studies as to where we make some decisions. Again, I would say start very early. If you haven't started and you're not very early, you can start now, that's fine. And you can ask some relatively simple questions about what's the target. So is that target on a dendritic cell? And you might want to apply some of these assays to understand how the dendritic cell's seeing your drug. Who are the patients? So there are differences in patient populations. It's not always straightforward. We used to say that if it's oncology patients, perhaps their immune systems aren't working so well, so there's a lower risk. If it's autoimmune disease, they're perhaps a bit overactive, so it's a higher risk. It's not quite that simple, but there are cases with specific areas of cancer where there is a much lower risk. So it is good to highlight. And for some diseases, there are specific HLA relationships, which again, you can target your assays to look at the HLA that are more enriched in the patient population. The other piece is the modality to be used. So we have lots of experience now with monoclonal antibodies. So generally, developability is good. We usually understand quite well the interactions they're making. But I know you guys will be working on very exciting bispecifics, trispecifics, even more complex molecules as well. So there are some uncertainties in those, and there are questions about, do you look at each individual binding domain? How do you look at the protein altogether? So I've not got details on that today, but certainly I'm happy to talk to you about some of those issues that arise. So the other thing is, when we ask these questions, we shouldn't really say, what's the target? Oh, great, it's not a dendritic cell, I'm fine. You might want to look a bit deeper. So in my experience, we find that often when we know where a target is, perhaps on a B cell, and we think we're dampening the B cell immune response, that we won't have a problem with immunogenicity. Unfortunately, sometimes that target is also on a dendritic cell, and you can see in the assays that it lights up really nicely. I haven't included it here, but we did have a nice paper where we used some of Proimmune's assays in 2017 on a molecule called ATR-107, and some people use that as a positive control in assays because it works really well. And that was designed to target B cells and dampen the response. But we had 75% of our phase 1 healthy volunteers generate an immune response to that drug, and the product was terminated at that stage. So just to get a bit into this target risk about receptor versus ligand. So if you have a choice early on, you might look at what's my risk of targeting that receptor. So in this case, the ATR-107 was an IL-21 receptor. So we're looking to block that receptor, but unfortunately, so that led to the uptake of the protein, which we could see nicely in the ProPresent assay. And there may be other ways, potentially less immunogenic, if you're targeting the ligand rather than the receptor. So this doesn't always hold true, but there's a couple of examples here. In the case of the IL-4 receptor, the MG317 product had higher immunogenicity that then prevented the efficacy, whereas it may be not so easy to target the cytokines, but in the case of the anti-IL-13, this is a lower immunogenicity approach. So what you can do with these is quite early on look at things like a dendritic cell activation assay or that internalization assay. You could use the ProPresent. It tends to be a bit expensive at the early stage, but if you're aware of what your potential risks are, you can make that as a targeted decision. So I wanted to give an example here as well. In the TNF inhibitors, there's, I would say, a higher immunogenicity than we would like to have for modern products. So they have been very successful, but we would expect generally to have lower immunogenicity with a new product. So what's been observed with these is if you, because TNF, the target is trimeric, and the drugs as monoclonal antibodies have their two binding sites, that you can get these complexes formed. And we were able to demonstrate with a ProPresent style assay the additional presentation of peptides when the target was included in that assay. So if you run these assays in a standard way, you don't necessarily put the target in, and you wouldn't see this kind of outcome. So it's worth thinking about what's actually going to happen inside the patient. The drug will be binding the target, and over time you'll get potential for these complexes to form. And in this case, the left-hand panel shows the kind of sizes that were formed, and the right-hand panel shows the increase in presentation. And you can see with the individual donors that there's a trend, and some of the donors go against the trend. So usually with these assays as well, we like to do quite a lot of donors, not just one or two, and think we've represented the outcome. But usually a minimum of 10, depending on the assay, it may be more informative to go to 30 or even 40 donors. I think we'll probably cover that in more detail as we go through the 2 days as well. So another area that's is always interesting from a project perspective, with our antibody drugs, we always want to have nice long half-lives. Can we extend that? So the technology is now for increasing binding to the FcRn receptor, so you get a lot more of recycling of your antibody. I guess it's a well-established strategy. One of the potential pitfalls in that is if, again, you're making complexes through binding of the drug to its target, instead of you getting lots of wonderful recovery of your drug, what can actually happen is the drug then, because it's in a complex, gets targeted for presentation. So in theory, FcRn binding or increasing FcRn binding lowers immunogenicity. In some cases, it increases it. And you can work this out from a risk assessment and some assays to look at that presentation level. You can also then look at, do I need to do something different with my drug? Perhaps the acid switch technology, where when you're getting into these compartments, you're releasing the target from the drug, can help with this. The other way is to look at with a one-armed approach, which kind of you can get in bispecifics, depending on what the combination of targets are. Sometimes bispecifics, you can actually make the problem a bit worse. So it is worth thinking about things like the pro-present type assay or an uptake assay to de-risk this early on. And it might be that you can use this with a tool compound rather than your candidate, so you can actually look nice and early. So thinking about integrating data. So this is quite a challenge. As shown earlier, the immune system is very complicated. We do have a good understanding of aspects individually. And one of the areas which over the last 20 or more years has really improved is this in silico piece. In this particular example, on the right-hand side, this is one of our favourite molecules, bococizumab. So it's an anti-PCSK9 antibody, which had about 50% ADA incidence and resulted in loss of efficacy that caused the program to be terminated in phase 3. I think I'll come back to this in tomorrow's talk as well. And in the example, we were looking at the various in silico methods and where they highlight the CDRs. So you can see the largely green bands of where the presentation occurred. And then the pro-present type assay is in that kind of pinky purple colour. So they don't always match perfectly, hence the need sometimes to do the assays. But you get a good idea from the in silico as to which sequences will get presented. I wanted to highlight here, there's a lot of data going into this. So this is like about 500,000 data points now going into making these models. And I think back in 10 years or so ago, people didn't like to use them because you'd get a lot more sequences predicted. So lots of false negatives. Whereas now they're really quite good at possibly overshoot. In this case, specific case, there was a bit of an under prediction. Moving on to the next one. So the other way people are using models is to try and combine different factors. in larger data sets. So in this particular case, published last year, some of my colleagues at the time at Genentech looked at a lot of the internal programs where you could see all of the data on the ADA incidences, and we could match up as well for specific individuals the types of co-medication they were on. And they were able to work through each of those dots as a cohort where they were making predictions on whether the ADA incidence would be higher or lower than 10% in a cohort. What information do you use to make this prediction? So nicely agreeing with the FDA guidance from 2014, they were not able to demonstrate very clearly with just the epitopes or the mechanisms of action, but once they started adding in the other factors, they became quite good at predicting whether something was going to be, I would say, low or higher immunogenicity. So I think this is a nice example of those risk factors that have been identified are really key, and you can take them into account. You don't need to go and do this or try and find this kind of data to see that these risk factors count. You should act on them now. So assess your projects, try and reduce these risks where you can, and you're more likely to end up with low immunogenicity. So this is a little complex. I won't run through it in detail, but some years ago we looked at how do we combine the data that we're generating. So we built a mathematical model where we looked at the dendritic cell component at the top, the T cell component in the middle, and the B cell component at the bottom. So this is a little bit like the cartoony diagram I showed earlier, only this would be a sort of mathematics or clinical pharmacology view of it. And what we were trying to do here was to identify, what are the key components. I mean, ultimately, we'd love to predict immunogenicity, but what are those key components that cause the immunogenicity that we can actually change? So we understand at the top left here, these yellow triangles are some of the highlights. So what's the danger signal in the system? So again, immunology textbooks will tell you you want to understand the danger signal and the foreignness, and this sort of adjuvant effect and the antigen effect are what combine to give you the immune response. So the model that we built helpfully highlighted that these are exactly the things you need to look at. So, and I think we'll hear more from Daniela as well about the importance of these kind of danger signals. So if you've got the assays that can measure and pick your molecules with low danger signal, that would be a good starting point. And then for the T cell response, that combination of the in silico with a T cell assay, such as the ProCern, will help you then determine what's my lower risk that I'm working to. So this is more complex. If you're interested in learning more about this, I'll be happy to talk to you about the whole modelling process that we have. I want to go into a piece on adalimumab. I'll touch on this briefly. So even back in 2011, the publication on the top right, we knew already anti-drug antibodies are associated with lower exposure and ultimately a loss of efficacy. So we have the dropout rate. It doesn't show so much sometimes in the first year. So a typical phase 3 clinical trial may not give you the answer to the long-term immunogenicity. And in this case, actually 3 years worth of data is where things really started to spread out. Again, very successful project. So some particularly executives think, we're not worried about immunogenicity because adalimumab had lots of immunogenicity and it was fine. So it had quite a bit. It's not really terrible, but again, for all the patients who come off it, it's not ideal. So what has also been demonstrated is the difference in the bottom right between just binding ADA and neutralizing ADA. So again, depending on your project area, this may be significant as to, are you getting binding that has relatively little effect, sort of affinity matured where you might have more direct impact because you're neutralizing directly, or a broader array of antibodies. So if you've got many epitopes, that will lead to clearance. So you need to take all of these things into account. But this is a good case study together with molecules like natalizumab, where we know the outcomes that occur typically for monoclonal antibodies. So we should pay attention. And the nice thing about adalimumab as well is we have all of the in silico in vitro data that we can generate on other products. So on the right-hand side, we have kind of the in silico. This was probably one of the earlier algorithms, so a lot came up. I've highlighted in red the sequences that the in silico would be, say, these are non-human. Even in a fully human antibody, the CDRs are typically a problem. And then how we would look at that with the PBMC type assay and a MAPS type assay to identify sequences that are going to cause problems. And this was followed up with a publication at the bottom where they actually generated T cells that would recognize these specific peptides. And there's a reasonable degree of concordance in there. It turns out that also in patients, there was a publication last year that highlighted a similar profile. In fact, I think there was an additional sequence that was found. So the take-home message from this slide is that on products we know that have immunogenicity findings in humans, we can find most of those liabilities through the in vitro assays. So if you can find them and then choose the candidates that don't have them, you're going to be in a much better place for the clinical trial. I'm going to skip that slide. What it really conveys is that if you want to be predicting immunogenicity, you need to run the assays, integrate the data, and you can run the simulations. The thing that we're often lacking is the clinical data on aspects other than the ADA. So that's really about getting these B and T cell measurements from clinical trials, which is somewhat challenging, but it does give you a richer data set. So I'll touch very briefly on, you don't really want to get to a point where you need these clinical mitigations, but there are options like looking at dosing, schedules, use of immunosuppressives in some areas. These are critical to being able to continue to use therapies. And then there's emerging opportunities around active tolerance induction. This is still quite early days of the science, but there's several different methods that people are trying to overcome immunogenicity for therapeutics. And with that, I want to summarize and say that the risk assessment really helps to develop successful medicines. You can integrate this different data from the different assays to assist in the decision making. And we can develop products with immunogenicity, but it makes sense to reduce your ADA liabilities and risks wherever you can. And again, you're going to learn in these next couple of days the different ways to do that and the kind of assays that you can apply to that. I think that was the end of my presentation. So thank you ever so much, and be happy to take any questions. Time.
Nice to meet you in person again in Shanghai. So we will demonstrate it. No? Okay. Yeah, Tim, I'm quite interested in B cell assays. You also mentioned in your slides. I know we currently know the well-established assays are more focused on the T cell activation. So could you say more about current B cell assays status? Are they validated or established assays can be used in science?
Yeah, so I think for this whole area, it's worth pointing out that validation is not complete. So when we talk about validation, it might be aspects of analytical reproducibility. And again, there the assays do have slightly different performances. They're somewhat noisy in places. Some of them are great, like the ProPresent is really reliable on same HLA type, same peptides presented. The B-cell assays, I think, are really challenging because the way that the B-cell response is working within the body involves the orchestration of the whole system. So the assay that we developed at Roche and published last year, it was very much kind of giving the system a hard kick and say, well, if there is a B cell that recognizes my drug, then we're going to find it basically on the assumption that we're kicking the system hard anyway. So there are a couple of more complex systems. So one that's emerging is a tonsil organoid system. So that seems to work reasonably well in generating vaccine responses. So where you've got a stronger antigen and adjuvant system. The other system that's used by the vaccine company Sanofi is something called Mimic, and they have quite an elaborate, workflow, automated workflow, which is essentially the same as I've shown in the cartoon. They take each of the systems and they need some good activation of the dendritic cells, and they have a way of doing that. And that's been shown to reproduce clinical trial data for influenza vaccines. It's just too cumbersome to use in preclinical development for biologics. And I think we'll probably hear from Daniela. So one of the issues we have is, we don't have standardized assays in a way where they're validated, where we know exactly what the outcome will be and we can make a very clear decision. We do need the teams working on each of the products to really evaluate the data for their context of use. Does that help? Okay, thank you.
Hello.
Hello.
I have actually two questions. The first one is about in vitro immunogenicity assays. I think probably each company has its own perspectives concerning which method might be the best. I think Eli published one paper concerning the CD8 deplete PKC silico culture assays might be the best. But what is your personal perspective concerning which methods work best in your case?
Yeah, so thank you for the question. These are, again, a bit of a challenge to say what's best objectively. We certainly see with a T-cell assay that CD8 depletion can help reveal some signals that with the CD8s present you would miss. So I would say it's usually better to see more and then make a decision on the more data and not panic too much if you see lots of signal, but really try and understand where that signal's coming from. So again, not just necessarily whole protein, but really looking at the peptides and saying, well, is the signal coming from a peptide that is something I can do something about? Do I need to do something about it? Because sometimes you see signals coming from the germline sequence, so we would not normally worry about trying to change that sequence. In the European Immunogenicity Platform, we've just had a paper out earlier this year trying to give some recommendations about these T-cell assays. I think there we did say that probably the CD8 depleted was a better standard assay to use. And we also, the key thing was the number of T-cells that were present in the assay, because the nature of the antigens, that there's usually not that many T-cells, or a low frequency, so you would normally want to look at about 2 million T cells per donor that you look at. So I'll be able to share that publication. I think it is fairly, it's an open access publication, so I'll be able to share that with the group.
The second question about the cascade of immunogenicity assays, and as you noted that the MAPS and DC peptide co-culture is kind of low throughput. And in your case, or Roche, for each project, how many molecules will enter into the MAPS? Because it's quite a good effort to do that. And do you really run these kind of assays for each project? Thank you.
Yeah, I mean, really good question. So at Roche, we took the view that we would carry out the risk assessment to determine whether it was worth us doing the other assays. So if you assess your project as being low risk and you run the in silico, and if there's not much there, the other risk factors are low, the maps, it's too expensive really. You know, even for an established company with the method established, it's just not worth it. So we would very much like select the projects on the basis of would we make a decision on that data we generate. I think as well, where I'm advising companies which may only have one or two assets they're taking forward, if you have like a platform approach and you need to use this platform elsewhere, it's worth getting this data early, because it might be that the first molecule you take to the clinic may carry some risk with it, but the rest of your pipeline, you've already assessed that risk and you're trying to overcome it. So if there is a problem, you're already aware and doing something about it. So I think practically the throughput, and we'll let Promune talk to this, might be like about 3 molecules for a run. In Roche, we kind of, when I arrived there, they reminded me that they'd invented the assay. So they were very proud that they could do 9 molecules. But again, we wouldn't usually use it for 9 molecules on one project. So we just ran the projects all together. I don't know if Nick wants to come in there.
Yeah, I mean, in terms of throughput, I would say we can run as many assays as you want us to, so that's not a problem. It's not a limitation. So typically, okay, per donor, there's a limit in terms of how many molecules you can put through. Theoretically, you can stretch that, but then, you know, maybe the resolution of the peptides will go down. You need a large cell number, basically, to work with, and that sort of accommodates maybe 3 to 5 molecules that we like to do. But you know, you can just run onto other donor sets. That's not an issue. And I think if you run a large enough cohort of donors, you get a representative sample of data, even if your donor cohort changes between projects.
And here again, it's an assay that's really good if you've got the same HLA type in different donors. You tend to see the same peptides, so it's less of a risk maybe than some of the other assays. Thank you.
I think Julia wasn't the pharmaceuticals. I have a question. Do you see the development, like we are all looking at helper T cells as an indication for immunogenicity? Do you think the development of some assays for regulatory T cells may help to kind of mitigate the risk more?
Oh, sorry, for which T cells?
For the
Follicular ones, yeah. So I think in terms of risk factors, we're probably okay with the current set of assays. For that truly predictive immune response and trying to piece together the whole system, it will surely be very helpful to have those follicular helper T cells. And again, this is probably where things have gone with the things like the tonsil organoids, that's really the intention, that you're getting a presentation and a recognition in a more physiologically relevant way. So we always are concerned with immunological assays when we're taking peripheral blood. Are these the real cells? You know, rather than the ones that might be within the lymph node, within the spleen, hidden in tissues. So there is just that practical element in terms of the numbers we look at, that we're often then restricted to using the peripheral cells.
If, for example, you have activation of regulatory T cells that may kind of mitigate more risk because it will suppress the immune system, and so I'm coming from a bit different point of view because.
Yeah, so the regulatory cells, I think, is an interesting area because so the big picture here is that we know there are regulatory cells that may already recognize some aspects of the therapeutic. We also may get them as a dampening down of a response after it's happened, but the instigation of that is going to be context dependent. So I think this is it's a challenging area to measure, but not impossible. And I think we don't do enough to measure that in the clinical trial setting so that we really understand what's going on. And I think one of the examples is with natalizumab, which with modern assays you see higher ADA incidences, but a lot of those go away. So, it may be that there's some more active tolerance going on. It may be something more passive. But yeah, we don't have the data as far as I'm aware. Thank you.
Thank you. Just to address, just to address the question that you just posed, I think that most people look at the Treg aspect, thinking a little bit about when we do mitigations, are we disrupting an epitope that might activate those regulatory T cells? And that is something that can be looked at in silico. I think that it is still kind of a nascent area. It's not as well developed as other areas, but it is an interesting aspect, right? You're trying to get rid of some potential epitopes. Are you now introducing or disrupting something that was acting as a regulatory type of epitope? I think that's something pretty worth considering as you look at the totality of the data.
Well, that was my question.
Yeah. Good.
One more comment. As Dr. Hill is a good question. You consider other immune cells like macrophages or the immune system design. They also might have some responses.
Yeah, I mean, that's a really good point. So I guess we've gone a little bit reductionist in this and trying to focus on key cells. And to the previous question, it's like, still, do we have the correct key cells? So I think with the macrophage, that a lot of what they take up is cleared without going through a presentation process. And our current understanding would be that the processes would be somewhat similar with the dendritic cells that we use. So as well, cautious that we're often using the monocyte-derived dendritic cells in these assays, and they will also be of a slightly different phenotype than if we used, I guess, the native dendritic cells from patients as well. So there have been some attempts to look at directly taking cells from the clinical trial subjects, and we do tend to see similar outcomes in the patient samples as we do in the donors, where you've got the HLA matching, and there may also be some other factors involved. But I guess, again, with any complex system, you want to have an understanding. Are you looking at enough of it to get a prediction of what the outcome will be? There's always the risk you might miss something. So if you've got a therapy that's looking to modulate the effect of the macrophage, you probably need to include some measure of, well, what are you doing? Are you dampening down the response? Are you increasing it? And does that have a knock-on effect for the immunogenicity?